Good afternoon, everybody, and welcome to today's Global Health Compound Design webinar. I'm very pleased to introduce today's speaker, who is Neil Berry. Neil is currently Head of Chemistry at the University of Liverpool, and he holds MChem and DPhil degrees from Oxford, and then moved to Liverpool in 2003, where he's been ever since. In 2007, he was appointed as a university lecturer and has risen through the ranks, and we can congratulate him on being made professor last year. He specialises in computational modelling to understand chemical systems more fully and predict molecular properties. And Liverpool is, of course, home to its own School of Tropical Medicine, so Neil has worked extensively on a number of global health projects, and today he's going to talk about two topics. The first will be about antimalarials, and the second about anti Wolbachia drug discovery. I'll hand over to Neil now to give his presentation. We'll take a couple of questions in the middle. We'll have a slight pause in the middle, take a couple of questions on the first section, and the rest of them we'll hold until the end. If you'd like to ask a question, please feel free to type into the chat box, and we'll try and get through as many of them as possible. I have my sidekick, Mark Gardner, keeping an eye on proceedings as well, because I have to confess that today my internet connection has gone down, so I'm relying on my phone to run this. This is going to be a first. So off you go, Neil. Over to you.
Thank you very much. So thank you very much, Caroline, for the introduction. Thank you to Caroline, Mark, for the invitation to give this Global Health Compound Design webinar series talk. So as Caroline outlined, it's very much in two parts my talk today. The first section is concentrating on anti-malarial drug discovery, and the second on anti-Wolbachia drug discovery. So an overall outline is just going to say a little bit about what our group do in a very general sense, and how we interface with lots of other experimental groups, both synthetic and biological. And it's really through the use of chemoinformatics that I'm going to draw out some of our key activities. As I say, it's going to be these two case studies I'm going to concentrate on. The first in the antimalarial drug discovery, which is exploring the MEP pathway. And the second part of the talk is looking at anti-Wolbachia, or A-WOL, drug discovery, where we've used ligand-based virtual screening combined with a high-throughput screen. So I guess our strategy within my group specifically is really to use this sort of simple molecular design loop to drive everything that we do. Obviously, what we do within our group is really the modeling, whether on the workstations or on clusters, and we collaborate with a wide number of synthetic groups, particularly Paul O'Neill's group here in chemistry, and others, but I'll highlight those. And as Caroline said in introduction, we have very strong links with a range of other institutions, including Liverpool School of Tropical Medicine, and one I'm going to talk about in the first part of my talk is Washington University. So really what we try and do in my group is use the chemoinformatics. So use the integration of chemical data with analytical and molecular design tools to identify best fitting compounds to address particular goals. So we're interested in doing predictive modeling. So we want to realize the next generation of chemistries that are going to do what we want them to do. And crucially, we also want to provide some chemical understanding. So what are the key molecular properties that give the outcomes that we see? So starting off with the first part, the antimalarial drug discovery exploring the MEP pathway. So for this part of the talk, I'm going to give a brief introduction about why we still need new drugs for malaria, briefly cover what the MEP pathway is and why we chose to target it, say a little bit about how we performed a high throughput screen And from that high throughput screen, we got some chemotypes, and there's one particular chemotype that I'd like to present to you, and I'll display that through the research aims, some synthesis and some structure activity relationships. And then that will lead into a discussion about the mechanism of the inhibitors that we found in this particular series. And towards the end, I will highlight a complementary approach of a non-covalent inhibition. And then I'll finish with conclusions and ongoing work. So I'm sure, as many of you know, clearly malaria is still a major global health threat. Huge number of cases and 405,000 deaths in 2018, and a massive human and socioeconomic burden with significant pediatric mortality. Plasmodium falciparum is the most lethal malarial species, and that tends to be the one that we focused on historically here in Liverpool. Current antimalarial therapies are failing, and there's increased levels of resistance growing throughout the world. And there aren't that many attractive drug targets at the moment. So on the right-hand side, we've got a picture of artemisinin, which is one of the frontline therapies, I'm sure as many of you know, but this is one of the ones in which resistance is increasingly growing across the globe. So our approach to this in this program was to target and exploit the MEP pathway. So on this slide, I outline what the MEP pathway is. And essentially, it's a biochemical pathway to generate isoprenoid precursors. So it's a pathway to generate precursors on the bottom right of the ChemDraw scheme, so IPP and DMAPP. And there are 7 different enzymes which take pyruvate plus glyceraldehyde 3-phosphate all the way through to these isoprenoid precursors. We like the look of this pathway as it was essential for Plasmodium parasites, but it's absent in humans. So for most of the enzymatic steps outlined in this pathway, inhibitors are known and are in the literature. So one of them I just want to highlight is the inhibitor phosphidamycin, which is an inhibitor of this ISPC enzyme. The structure of phosphidamycin is on the right-hand side, and this demonstrates the safety of targeting the MEP and validates the viability of MEP as a target for antimalarial drug development. So we believe that targeting this pathway is a good choice because it's essential to the parasite. It should have some selectivity as it's exclusively in the parasite and not in the human. And with the evidence of phosphidamycin, we believe that it can be druggable. So the stage of the pathway that we are focusing on is the ISPD part of the pathway, indicated in the red box. So that catalyzes the condensation of MEP and CTP to give us this molecule here, CDPME. Very few substrate competitive inhibitors have been developed. There was very little in the literature when we started. No potent Plasmodium IspD inhibitors reported, and so we wanted to target this for compound development. So our initial approach was to carry out a high throughput screen, but a selective high throughput screen. So we used the biofocus library of about 500,000 compounds, and we identified 5,000 compounds that were going to be tested in the in vitro assay. The way we identified those compounds was by using molecular similarity through fingerprint searches of compounds which were either similar to the substrates or the products of ISPD, or were similar in some way to to structures which have been shown to have some activity in other stages of the pathway. In addition to that, we also searched for molecules which had bioisosteres of phosphate groups somewhere in them, because in the structure of either the substrate or the product of ISPD, phosphate recognition was believed to be important. So we had an element of similarity to a range of queries in order to identify compounds which would go into our in vitro screen. Complemented to that, we built an homology model of Plasmodium IspD, and we docked multiple thousands of these compounds into the binding site, and we selected those which looked some of the best fits. So through the cheminformatics of the fingerprint searches and the docking, we selected 5,000 best looking compounds from our 500,000, which went into the in vitro screen of potency identifying actives. The next stage we did was to take what we knew from that screen. So we'd screened 5,000 compounds. We had some which were hits, and we had a large number of misses. And again, we used some cheminformatics in order to do some machine learning and to score and rank all the other compounds that hadn't been screened to date. So by using our knowledge of the chemistries and the molecular properties of the active compounds from the first screen, we went back into the biofocus library and selected those molecules we believed had a very strong chance of showing activity. So we selected another 5,000, which went into the in vitro high throughput screen, and we then identified overall 54 hits through this screen, 10 potential chemotypes, and the one chemotype I want to talk you through for the rest of this first part of the talk is the lead hit, is shown on the right hand side, which has an IC50 of around about 480 nanomolar. So what we want to do was to take this hit and to move it as far as possible towards a lead candidate suitable for clinical development as an antimalarial targeting ISPD. So what we're going to focus on is looking at the hit structure. So it's a benzoisothiazolone. So it's going to be called BITZ for the rest of this talk. And we're going to explore some of the side chain modifications in order to develop some SAR and the chemotype. We want to enhance the efficacy of the hit, and you want to optimize some of the drug-like properties. And we want to try and probe the chemical space of the enzyme target. So again, on the left-hand side, here is our hit molecule. And as I said, we want to develop some SAR, understand what is the optimal requirement functionality in order to drive this IC50 further down. And in summary, what we knew at the start from the high throughput screen is captured in this diagram at the bottom. So it looks as if this core was essential. So the benzoisothiazolone core is essential. This C ring, if we remove the sulfonamide, that promotes activity. So removing that equivalent group from our hit. Direct N-aryl attachment is optimal. Alkyl or heterocycle seem to be inactive. In terms of the connectivity of the C ring, meta substitution seemed optimal. Ortho and para was not tolerated. And we had a question of what is in the optimal D ring. Is the aryl ring a polar heterocycle? And how about the substitution on the D ring? What might be the best there? So I'm just going to highlight a small number of compounds, but I'm only highlighting a small number here of the overall project. So saying something a little bit about the synthesis of these molecules. These molecules can be synthesized fairly readily But as you can see, it's quite a linear approach. So starting off with this carboxylic acid on the top left, standard thionyl chloride to produce the acid chloride, condensing with the amine to produce the amide, and then we can oxidize up the sulfur to a sulfoxide using sodium periodate, and then we're able to couple in via a palladium catalyzed coupling a range of different aryl groups, as long as the boric acids were available, and that usually proceeded in good to very good yield. And the final step was this ring closing step using thionyl chloride to form that 5-membered ring and that nitrogen sulfur bond. So again, just highlighting just 3 key compounds here. So we had a variety of R groups on the para position of the D ring. So electron withdrawing, electron donating, and also sort of a halogen. So we can see that the IspD inhibition, we've increased the in vitro inhibition, in some cases really quite markedly. The whole cell growth inhibition is okay, but it's still really could benefit from being even lower. The one thing that we did notice was that the calculated C log P are really quite high at the moment. So one effort to try and address that high C log P, was to have a look at removing the terminal aryl ring and introduce some rings and some functionality with polarity in. So to work through the synthesis here, we can start off with this nitroaromatic, which has a bromine group attached to an sp3 carbon, and with a suitable nucleophile can be displaced by the amine, and then the nitro group can be reduced down using hydrogen palladium over carbon to produce the amine, coupled in to the acid chloride, and then ring closed to give our benzoisothiazolone core, our aromatic ring, which is meta-substituted, but this time just onto an alkyl polar group. So as you can see, we have 3 compounds shown here, where X for the oxygen, or CHF, or methylated nitrogen. So the IspD inhibition is, again, it's still in the sort of the hundreds of nanomolar, but unfortunately we seem to have lost a whole cell growth inhibition, but C log P has come down a little bit. So in terms of potential mechanism of inhibition, we built a IspD homology model based on the E. coli IspD protein. It's got the PDB code there. So from our modeling, homology model, and from the docking, we believe that these benzoisothiazolones have a similar binding alignment to the natural product CDP-ME. So on the left-hand panel, we've got CDP-ME in the active site of IspD. In the center panel, we have inhibitor number 3 docked into the same area, and you can see its nearest neighbors in terms of the residues in which this compound is interacting are very similar to CDP-ME. But after the shape recognition of this inhibitor number 3 being recognized by the binding pocket, we believe that there's a crucial covalent adduct formation where the sulfur from this cysteine 202 actually attacks the nitrogen sulfur bond and breaks open the ring. And on the right-hand panel, we've got inhibitor 3, which is covalently bound, and we can hopefully you can see the sulfur-sulfur bond being formed there. So some evidence we have for believing that's a mechanism for action is shown through some chemotype validation work if we look at the table, here we have our very simple inhibitor, which only contains the benzoisothiazolone core, just with an aromatic ring on. And that has some activity. It's not great, but it has some activity. If we take out this nitrogen sulfur linkage and we just look at this second compound, then we see no inhibition whatsoever. And if we expose the IspD enzyme to the compound at the bottom, where the ring has already been opened, and you can notice the similarity in terms of the position of this sulfonamide group compared with the initial HTS hit, we're not seeing any activity either. So added on top of that, we undertook some site-directed mutagenesis work to confirm the involvement of the cysteine residue in a covalent interaction. So this is work which took the E. coli IspD analog and sensitized it to the benzoisothiazolones. So wild type E. coli lacks the cysteine 202, and as you can see on the bottom left, that has an IC50 of over 100 micromolar. If we introduce the alanine 14 to cysteine mutation, then the E. coli IspD is now susceptible to benzoisothiazolones, so at the level of 450 nanomolar. Equally, we showed it's possible to desensitize the Plasmodium IspD to BITZ. So we introduced the reverse cysteine to alanine mutation, and so that had an IC50 of 470 nanomolar, and we replaced the active site cysteine residue. So we can see that by changing the cysteine to alanine, we have desensitized the IspD. So just a very short summary of some of the work we've done on this. We've sort of shown that we've got a new chemotype active against Plasmodium IspD enzyme. There's a large number of analogs have been synthesized of the order of well over 100, and obviously I've just shown a few here. The most potent IspD inhibitors to date, so the most potent one is down at level of 70 nanomolar against the enzyme, and whole cell growth inhibition is of the order of half a micromolar. We have rationalized the covalent mechanism of inhibition, but we are aware that covalent modification can be a risky strategy in lots of ways. So we've managed to take this lead hit, which looks like this, and we've gone down to this sort of molecule here, and we're still working on these. So alongside looking at the benzoisothiazolones, we also undertook some work looking at this this structure on the top right-hand side. So this structure actually also inhibits Plasmodium IspD, and it came out as we were working on this enzyme. So it has low nanomolar activity, and it first recognized through a phenotypic GSK screen. So hence it's got an MMB number. So the most potent inhibitor found at that stage has this kind of SAR. So we've undertaken SAR development around the A and B ring here. Stereochemistry in the C ring here has been shown to be important. We have some understanding about the functionality that's tolerated on this top carbonyl group. It looks as if this secondary amine is required, and we've undertaken some SAR development on the bottom right-hand side. So this is a complementary strand to the targeting IspD, and it's a non-covalent mechanism of action, and it's been shown to be competitive with CTP. So we've published on this, and the reference is at the bottom there. So overall, we've targeted the MEP pathway and the IspD enzyme within that. believe it's a good target to antimalarial drug discovery. We've identified both a covalent and a non-covalent inhibitor. It arrests the MEP pathway through IspD inhibition, and we're proceeding with 2 complementary inhibitor chemotypes, expanding SAR and optimizing the PK. So this first part of the talk was conducted with Professor Paul O'Neill as main collaborator in Liverpool. The lead collaborator was Audrey Odom from Washington University in St. Louis. And I'd like to acknowledge the PhDs who worked on this. So Catherine Price, Natalie Roberts, Matthew Pye on the synthetic side. Then Alex Lawrenson was a PhD who helped out in the modeling. Raman Sharma, PDRA on the modeling, and Chandra was a PDRA on synthesis. So that's where I propose maybe I can pause at the end of the first part of my talk, and I'm very happy to try and answer any questions at this stage.
Right, so if anybody has any questions, please type them into the chat box. I know Mark has one ready to go whilst you're doing that. And we'll just take a couple now and then we'll take the rest at the end. Mark, would you like to ask your question?
Hi Neil, really nice talk. I was just going back to the hit finding, and you know, I really liked your approach of using several in silico methods for the finding the hits. I wondered, you know, is it possible to say which of them were most or least successful in this particular case?
So I think as ever, from what I recall, there wasn't one which was completely outstanding over another. So I'll have to go back and remind myself of the exact numbers, but there wasn't, as I say, there wasn't a particular fingerprint, or there wasn't a particular metric, or a particular, or the docking wasn't much better than anything else. So I guess our approach is always to try and sort of take the consensus approach. So I think that's where we are with that.
And actually, I was interested in your machine learning models. What sort of methods did you use, and did you use those models to decide on candidates for synthesis as you were going through the project?
So the models that we used were categorical learning models, so sort of an active, inactive, categorical models. And so I'll touch on this maybe in the second part of the talk a bit more, but essentially we used sort of molecular fingerprints, some molecular properties, random forests, support vector machines, and neural networks to try and identify compounds which were consistently predicted as likely to be active. And again, taking this sort of consensus approach that we had 3 different modeling approaches: random forest, support vector machines, and neural networks. If they all predict something is likely to be active with 90% confidence, then we're going to have a lot more confidence in that particular molecule than a molecule which only is supported well by 1 out of 3 methods. But as I say, that I'll expand on that a little bit in the second part of the talk. Right,
great. So I think probably, if there aren't any other questions from the audience at the moment, do you want to move on to the second part, and we'll get to those at the end, right?
Great. Okay, so the second part of the talk is focused on anti Wolbachia drug discovery. So using ligand-based virtual screening in combination with the high throughput screening. So what I'm going to cover is what filariasis is, how Wolbachia fits into this story, and why we're targeting Wolbachia. say a little bit about what the current therapy is and the issues with it, say what we're hoping to achieve here, and then I'll walk you through an iterative high throughput screening campaign which has been led by virtual screening. So the idea is that we're going to use virtual screening in order to select compounds from an HCS deck, which can then be assessed, and then hits can feed into the research program. So filariasis is a parasitic disease caused by an infection from roundworms. Very serious health problem, so it can cause lymphatic filariasis, as shown on the left-hand side, or onchocerciasis, otherwise known as river blindness. So as you can see, there's well over 150 million people who are affected by this, and it's a leading cause of global morbidity. So our approach is actually to target Wolbachia. And Wolbachia is a bacteria which lives in the gut of the filarial worms. And Wolbachia itself is essential for these worms, in that it's essential for larval development, embryogenesis, and also adult worms without Wolbachia die quicker. So the Wolbachia is actually has a symbiotic relationship with the filarial worms. So if we can target and eliminate in an elegant way the Wolbachia, then the worms should not cause the problems that they currently do. So drugs which target Wolbachia will be prophylactic, should be transmission blocking, and should be macrofilaricidal. This should all help in terms of clinical case management. So the current therapy is this molecule on the right-hand side, but involves 4 to 6 weeks of doxycycline. This sterilizes adult worms, it blocks transmission, kills adult parasites, and improves pathology. However, it does have issues. It's a relatively long treatment, and so there's issues due to compliance, and it is contraindicated in pregnancy and children. So the overall goal of the anti-Wolbachia program is identifying novel therapies. So what I should say here is what I'm going to touch on in this talk is only one small part of a large program of anti-Wolbachia drug discovery. And it will become clearer as it gets towards the end of the talk. So the global aim is to find treatments for targeting Wolbachia, suitable for mass drug administration in a reduced time frame, hopefully about 1 week compared to up to 6 weeks with doxycycline, safe for pregnant women and children, hopefully minimize resistance, usable in areas where lower is co-endemic, improve morbidity management, and with the end game of elimination being the ultimate goal. So one of the earliest things that was performed in the A-WOL program was a screen of 10,000 compounds from a biofocus library. So in summary, start off with 10,000 compounds. That went through, at that stage, what was really quite a laborious screening mechanism. Since then, the technology has improved a lot and the throughput has improved a lot. But at that time, this was a huge amount of work. Went through hit confirmation, hit validation, and end up with 50 hits. And so in the next couple of slides, I'll say a little bit about how we looked at those 50 hits to identify 6 diverse series. So the 6 series are down on the bottom, and I'll highlight this one on the left-hand side because I'll come back to that towards the end of my talk. So just saying a little bit about how we had a look at the 50 hits from this A-WOL1 screening of 10,000 compounds. So we clustered these compounds just using some molecular fingerprints and used principal component analysis just to see how they sat in some chemical space. And these circles are indicating the clusters of compounds. We also wanted to have a look at in a different way. Were there some clusters which were much better than others? And so we looked at using some Pareto selection criteria in order to find the optimal balance of properties. So obviously potency is a key and necessary part of any potential drug molecule, but obviously we want to balance that against other properties such as ligand efficiency, such as physicochemical properties such as solubility and log-P, such as toxicity and ADME. And so we had a range of different properties that we were interested in finding the most, which molecule or molecules had the best overall profile at this stage. So having done that, it actually turned out that the optimal region in this particular view is in the top right-hand corner of this view. And this actually contained hits from all 6 series. So at that stage, we made a strategic decision that while ultra high throughput screen was being developed with AstraZeneca, we would use chemoinformatics to inform screening priorities of the MMV compound library that we had in the School of Tropical Medicine in Liverpool. So obviously we had all the data from the initial 10,000 compounds, so we all had hits and the misses, and we wanted to use that information similarly to what we did in the IspD project to learn what we could from that in order to make the best informed selection of compounds. So what this program from the chemoinformatics side was about was selecting compounds to expediently expand SAR around the series we knew, around the 6 series we knew, perform some hit expansion, especially around some interesting looking singletons, maybe achieve scaffold hopping to new chemotypes, and we wanted to probe areas of chemical space previously not sampled. And so our way of doing this was having iterative rounds of chemoinformatics. So graphically, what I'm trying to show here is that in our first step, we looked in the 500,000 compounds in the MMV library, and we used similarity searches to any hits from the 10,000 A-WOL1 screen. That took us down to about 17,000 compounds, which were cherry-picked, went through the screening triage, and at the end we had 36 good-looking compounds. So just to say what this 50 and 90% mean, this is 50% or 90% with respect to our gold standard of doxycycline. So we had 36 hits, which were at least 90% as good as doxycycline. So just to put a little bit more information about the chemoinformatics, we just did some fairly simple similarity searches using a range of fingerprints, as indicated here, using the Tanimoto coefficient, but we did explore a range of different data fusion methods in order to work out which might be the most consistently predicted compound to enter into the actual screen. So we used some simple sum of scores. We looked at sum of ranks, we looked at reciprocal ranks, and this other scores Z2. So we managed to, taking this approach, we managed to increase our hit rate to about 2%, which is about a 4-fold increase on the hit rate from the A-WOL1 screen. In terms of how we did this, we used data pipelining through Pipeline Pilot quite a lot. And what I'm going to come on to next is how we've used R and one particular package within R known as Carrot to do machine learning and compound selection. So our second screen was to take everything we knew up to this point in time. So everything from the A-WOL1 10,000 compound set, everything from the 17,000 screen 1 we've just been through, and build some categorical models, both using random forest support vector machines, and we use Carrot to perform some hyperparameter optimization using a multiple cross-validation approach repeated many times. In terms of descriptors, we use some which captured the molecular properties, so things like log-P, hydrogen bond donors and acceptors, also fingerprints. So we believe that we found some robust models with the area under the receiver operator characteristic curve of more than 0.7. And again, we looked at a range of different data fusion methods. So we built models which we had confidence in. We went back to the library and applied those models to the 500,000 MMV library to select the next tranche of compounds, which is 14,000 compounds, to enter in the high throughput screen. They were then screened, and in this case, we end up with 100 more compounds and 90% hits, at least 90% as good as doxycycline. In terms of the results, we believe that this machine learning was pretty effective. We had a 3.5-fold increase in hit rates compared with screen 1, looking at a 90% doxycycline hits. The random forest approach greatly increased the hit rate, but mainly expanded SAR, whereas support vector machines had a poorer hit rate compared with random forest, but does seem to sample new areas of chemical space. So this can be shown in this PCA plot of chemical space, where we have the hits from random forest in green, hits from similarity in blue, and the SVM hits in red. Another piece of evidence to show that we believe that this chemoinformatics selection was worth doing is that plates which have no hits on actually had no hits on when it came to it. So in terms of expanded SAR, we've managed to expand SAR around some chemotypes in the biofocus screen. So we can see in this table that this chemotype number 1, we screened a number of molecules to expand the SAR, and we've managed to expand the SAR both in a very positive sense, the 90% doxycycline threshold, but also a large number of negatives. And we've found some novel chemotypes, so over 10 novel chemotypes have been detected. Most of these are currently singletons, but some are present in some numbers. In terms of probing chemical space, if we compare in red was a 10,000 biofocus A-WOL 1 library. In blue is our first compound selection, and our green is our second compound selection. So I think even though we're looking at a fairly small percentage of the overall variance in this PCA plot, hopefully I think you'll agree that we're moving into new areas of chemical space, particularly with this arm of green down here. So we did one more round of compound selection. So our third chemoinformatics machine learning compound selection. Again, this is using categorical modeling. Keeping our random forest and support vector machine, we added onto that neural networks. We did the hyperparameter optimization in the same way. Same range of descriptors we used. We only used models that we believed looked robust. But in this particular screen, we paid attention to applicability domain of the model. And I'll say a little bit more about that in a minute. Interestingly, there's a lack of correlation in predictions between these three approaches. So we think that provides extra evidence of the benefits of taking this, this approach of using multiple different inputs in order to build up confidence in a particular prediction. So just coming back to this idea of the applicability domain. So I guess whenever we learn a machine learned model, that model is not going to know about all areas of possible chemistry. And so when we built the models in this third compound selection, we kept track of the types of chemistry in the chemical space which these models were built on. And the prospective molecules were assessed for closeness to the training set. So what I'd like to highlight is one or two entries in this graph below. So we can see that, for example, the random forest so that's within the applicability domain, both the 50% blue and the 90% red are higher than the random forest compounds which are outside of the domain. The same is true for neural networks, in that sort of, I guess, and that is what we'd expect, in that when we have a model which we have confidence in, we believe that it's going to know more about the chemistries which are close to the molecules it's seen before, rather than molecules which are far away. Interestingly though, support vector machines did not show that. In terms of chemical space of the hits that we identified, we calculated some physical chemical properties of features of the hits and looked at in PCA space. And in black, on the bottom left, we can see the whole MMV library. Red are the screen molecules, and you can see the hits are really concentrated in one area of chemical space. Looking at some physiochemical descriptors, here we've got polar surface area and A log P, and also molecular weight against A log P. And there seems to be a sweet spot in terms of physiochemical space for identifying hits. So as I said, all that screening activity was happening as other arms of the A-WOL program were progressing. So there's the A-WOL arm, which was developing the ultra high throughput screening with AstraZeneca. But alongside that, there was medicinal chemistry, I guess, hit to lead optimization. And that happened on all the chemotypes. 6 chemotypes came out of A-WOL1, but our leading chemotype was this chemotype here. So this came from the screening hit. It developed through various iterations, as you can see. Oh, sorry. We moved from a thionopyrimidine to an azoquinazoline, so we did a scaffold hop. It became clear at a very early stage that this particular arrangement of the CF3 group aromatic ring and amine was important. And we looked at a large number of substitutions at this 2 position down here, and identified this compound AWZ1066. So huge amount of medchem optimization occurred both in-house with industrial partners, and we had good translation between in vitro and in vivo Wolbachia activities, and that's moved to candidate selection now, and that's detailed in this PNAS publication. So we've gone from this hit to this candidate. This candidate matches our target candidate profile, can deliver 90% Wolbachia reduction in 7 days, compared with doxycycline in 28 to 42 days in animal models, has a faster kill rate, seems to be highly specific against Wolbachia, no effect against lower, and is in preclinical development and expected into first into human in about 12 months. So as I said a couple of times, I guess what I presented from the cheminformatics side is just a small part of the whole A-WOL program. And the A-WOL program initially started off in the School of Tropical Medicine, and in 2007, this is pretty much what the portfolio looked like in terms of Wolbachia therapy. But over a huge amount of effort from a huge number of institutions and people, we now have a portfolio which looks more like this. So lots of credible chemotypes, several lead series, plenty of candidates, some moved proof of concept drugs, and I guess some highlights are the AstraZeneca ultra high throughput screen. It's been performed, so screen their whole deck and have a huge number of hits. Lots of lead optimization projects, 2 candidate drugs in preclinical and 2 repurposed drugs. So I guess on the ligand-based virtual screening, I think we showed you delivered an increased hit rate using our iterative cheminformatics approach, expanded SAR around hits, discovered novel hit chemotypes, and provided a profile for physical chemical properties for active compounds. Ongoing work in particular is the first in human trials in the next 12 months of 1066. So there's a huge number of people that have been involved with this, but I particularly want to acknowledge from the Liverpool side, Paul O'Neill, Jacqueline Bibley, who did all the cheminformatics and machine learning, and David Hong, who provided a key contribution in terms of the medchem optimization. In the School of Tropical Medicine, there's Steve Ward and Mark Taylor are the PIs on that side, but a large number of people, and I highlight here really the key screening team. In terms of the A-WOL program, you can see there's a large number of people who have been involved, but a huge amount of thanks needs to go to the Gates Foundation for awarding the grants to us in order to perform this optimization. So I think that's where I'd like to leave my that section of the talk, and again, very happy to take any questions, and just want to say thanks again for the opportunity to show this and share this with you.
Thanks, Neil. That was great. Lovely to see someone go through all the different types of machine learning models. You just answered my question perfectly. It's very nice to see the difference that you got from each one of those, and to see a nice proof of ligand-based virtual screening rather than docking-based virtual screening. You're actually doing fingerprint methods there. Do you think you would be better? Do you think adding any sort of 3D information to that would have made for better models?
I suppose we should try and sort of test the hypothesis. I guess there are several papers in the literature I'm sure you're aware of that sort of, certainly on the data sets that they've looked at, there's not compelling evidence that just throwing three-dimensional information in without any specific information actually increases hit rate. If we had let's say we had a crystal structure of what the main target might have been in the A-WOL package, then absolutely I think we should throw some three-dimensional information in there, whether it's in terms of pharmacophores or docking. But given that we don't know the actual enzyme or whatever it is that these things are targeting, my feeling at that stage was it would more likely just increase the noise with not much benefit to the signal.
Okay. We do have some questions in the chat box, and please feel free if you want to ask questions about the first section of the talk. That's absolutely fine as well. The first one here is from Peter and Jogo, who says, what kind of software programs are used in machine learning and similarity searches?
So for the similarity, we were using Pipeline Pilot from BioVir, which we've had sort of found very good, very quick. It can sort of cope with huge numbers of molecules, and it does things very nicely. In terms of,
you could of course use NIME as well, couldn't you?
Yeah, absolutely. And in terms of the machine learning, we were using our statistical package, but mainly this package Carrot, which I highlighted in the slides. So there we are. So Carrot stands for something like classification and regression training, and is a really good, I think it's really good sort of all-encompassing package, because it provides a sort of unified approach for just saying here is my data set. I'd like to try and find the best model you possibly can using a range of up to, I think it's got about 250 plus modeling sorts that it knows about, and it will do all the hyperparameter optimization and training test set splits, and it does things very nicely in our hands. Yeah, and so obviously that's open source and available and works very well.
Just out of interest, can you use that from within NIME?
We haven't done that, but I'm pretty sure you should be able to, because I think we have on other projects we've used NIME and then interfaced with R, and so I don't see any reason why that couldn't work. So you could imagine using NIME with RD kit and carrot in some way.
Yeah, I wondered that. Yeah. There's a question from Camilla here who says, did you use chemical rescue with IPP to identify the hit? If yes, was it ammonium salts? Do you know if lithium salts would do the same?
Okay. Yeah, so we did do some chemical rescue experiments. I can't remember the precise details, but they'll be in the paper I highlighted. So I think the information should be in that paper from in ACS infectious diseases, I believe. And if it's not, please feel free to contact me.
I'll look there, she says. Thank you. Yes, and just to say that the session is being recorded and it will appear on the MedChem Clips YouTube channel, and you can always find the recordings for this webinar and all the previous webinars by looking on the MMV website. You need to look on the computational chemistry pages, and they're all listed there. There's lots of interesting talks there that you may be interested in. Any more questions from anyone? Time for a few quick last questions. Actually, in the first part of your talk, you started your campaign looking for phosphate mimics, but you ended up with compounds that really didn't look as if they had aromatic rings in what was the phosphate binding site. So have you got any comment on that?
Yeah, it's interesting. We sort of wondered that as well. I guess one possible rationale of that is that because it's an enzyme performing a bond cleavage and bond forming, then actually you want recognition of phosphate, but you don't want it to be bound. So maybe there's an argument there about sort of, for an inhibitor, you want it to bind really quite strongly, but maybe there's difference between sort of recognition and inhibition via binding in some way.
Yeah, I wanted that. So effectively the binding is being driven by the covalent bond condition.
Oh, Neil, I did have a quick question, if you've still got time, about the trifluoromethylpyridine in the second talk. I suppose it's not what everybody would reach for first. Did you look into? It may be well known. I don't know. I don't know it though. Did you look into what might be going on there? I can see it's an evolution of the phenol, I suppose. But
yeah, so lots and lots of things were tried around that ring, and this just seemed by far and away the best thing.
Potency or ADME properties?
At that stage, it was potency.
Yeah.
Yeah, and I guess sort of once we were fixed into that, but as you can see, we did sort of introduce nitrogen at this middle structure here. I guess with a view to, we were obviously always fighting this battle, as is so frequent, to try and keep the log D down in sort of some kind of nice space we wanted. So introducing that nitrogen in that position did help. But in terms of sort of in-depth understanding at the moment, I'm not sure I've got any particular insight on that.
Thanks.
Okay, well, I think we've reached the hour now. I'll say a huge thank you to Neil for a great talk and some really interesting techniques that you've outlined there that I hope people will be able to use in their own projects. And with that, I think we'll close the meeting. Thank you everybody for attending, and look out look out for announcements on the MMV website. We'll send around some emails as well. Okay, thank you, everybody.